Hello my champions, welcome you all to 360 out of 360 in NEET Bio within 250 days series, and today we are going to talk about AIDS. And AIDS is caused by bacteria? No, it's caused by virus, and we spoke about it when we were dealing with bacterial, viral and all these diseases. If you have not watched that video and you think it might be useful for you, then go give a watch. And what did we study previously in the previous day? We spoke about acquired and innate immunity. We spoke about active and passive immunity, vaccination, immunization, allergies, autoimmune diseases and all the lymphoid organs which are there in your body. And if you want the PDF for the same, you can join our WhatsApp channel. It's just for us, the family of NEET people. Yeah. And were you able to solve this question? Well, those of you who have attended the class would have surely be able to, and if you are very new, then also you can. I'm pretty sure about that. Just have the confidence and read it very clearly. Antivenom injection contains preformed antibodies. Antivenom as in, for example, some snake bite. Okay, some snake has bitten us. At this point of time, we do not wait for our body to produce antibodies against it. Instead, we give preformed antibodies which is already there. You just inject it so that our body can be saved, right? Now, while polio drops, when you talk about polio drops that are administered into the body, what does that contain? What does that contain? You know there is polio vaccination. So they are asking you, what will it contain? Gamma globulins, attenuated pathogens, activated pathogens or harvested antibodies. Basically, it contains attenuated pathogens. This is an example of vaccination, children. This is an example of what? Vaccination. And in vaccination, what do you give? You give attenuated antigens. You give attenuated, that means weak pathogens which have some changes in them such that they can only ignite an immune response, but they won't cause you a trouble. No trouble, no sign of infection would be caused by them. So that is why we say attenuated pathogens. They are weakened pathogens. They are not in their full vigor to cause the disease. Okay, I hope that is understood. Activated pathogens obviously you cannot give. You don't want to kill yourselves, right? So in polio drops, which is a vaccine, you are basically giving attenuated pathogen, and this came in the year NEET 2016. Yeah. Going ahead, children, the number of questions which were there for this chapter has been increasing as I am telling from day one, but it has been repeated because we never know, some new child might be attending this class. So yes, this chapter has been increasing the number of questions each year. So we never know, this year it could be more also. So yes, let's start with our today's topic, that is AIDS. And for the first time, I want you to read through a topic and then we will be seeing one by one what we need to study. Okay, so let's start. The word AIDS, okay, the word AIDS stands for acquired immunodeficiency syndrome. Before that, I'll show you that we have to fill this column. So we have to fill the column of what is the name, what does AIDS stand for, and how is it spread, and who are at risk. Okay, so after reading this whole passage, we will be able to understand that. You have to find the answers. So the AIDS word stands for what? Acquired immunodeficiency syndrome. What do you mean by acquired immunodeficiency? You are acquiring it through your lifetime. It's not like you are getting it from birth. Okay, that can happen when the parent is having AIDS. That is something different. But this is something that you are acquiring. And what happens during this? Immunodeficiency happens during this. So basically, you become deficient. Your immune responses would be very slow. It will be really bad. Okay, there won't be quick responses, or probably there could be lots and lots of other trouble. For example, I might get a fever, but this fever, which usually subsides within 1 to 2 days, might not subside. So that is the problem. So basically our immune system gets very weak. So this is acquired throughout the lifetime, and children, basically it's a syndrome. What do you mean by syndrome? When the word syndrome comes, that means it's a so many bunches of symptoms. So many symptoms are coming over here. So this is acquired immunodeficiency syndrome. That is, throughout your life you're acquiring some immunodeficiency symptoms. So many symptoms are coming in. Okay. Now this means deficiency of immune system acquired during the lifetime of an individual, indicating that it's not a congenital disease. What do you mean by congenital? As in, it's not a genetic disease. Okay, it's not a genetic disease. Now, syndrome means a group of symptoms, as I already said. Now, AIDS was first reported in 1981. It's an important year to remember. Please remember it. And in the last 25 years or so, it has spread all over the world, killing more than 25 million people. Now, it would be more because obviously this NCERT book is also old. So, it would be obviously more. But yes, it was discovered in the year, or basically first reported in the year 1981 in US. In US it was reported first, but it is said that this particular disease happened to occur in Africa first and then probably would have travelled down US as well. Okay, and it was from chimpanzees that we got it. Okay, that is also a piece of additional information. So what do you need to remember? 1981, children, will you forget this? Please do not forget it. Okay, now yes. Okay, AIDS is caused by the human immunodeficiency virus. What is the causative agent? It is the virus. And what is the name of the virus? HIV, human immunodeficiency virus. So HIV. Okay. Now, a member of a group of viruses called retrovirus. Now, this HIV virus, it comes under a category of viruses which you call as what? Retroviruses. Now, retroviruses are very unique. Retroviruses are very unique. You will get to know why later on when we talk about their action. How do they work? Now, okay, so retroviruses which have an envelope enclosing the RNA genome. That means instead of having a DNA, they have an RNA as the genetic material. Was it? They have a RNA as the genetic material, and also their body is enveloped, as in they might have something that covers up the whole body. It's just like how we have a membrane, they also have an envelope around them in which they have the, the genetic material, which is an RNA. Now, we'll get to see the structure fully, okay, when again, as I said, when I talk about the mode of action. As of now, we have to fill the column where we have to talk about all those, how it is spread and things like that, okay. Okay, transmission of HIV infection generally occurs by sexual contact with infected person. So, there is a person who is infected. Now, if you are involved in sexual intercourse with the person who is having HIV, you can also get this particular disease. Now, how does it spread? With this information, you get the idea that with the help of fluids, the body fluids, it is spread with the help of body fluids. For example, you might have genital fluids, okay, the semen, the vaginal fluid, everything could be a part of it. So, one reason is that genital fluid is there, which is why sexual intercourse could lead to this particular disease. Next thing, transfusion of contaminated blood and blood products. For example, a person is having HIV again, and now this person does not know about it, gives blood to another person who is in need of it, and during this process screening has not been taken place. Screening of blood has not been taken place. At this point of time, obviously when it is going through blood, it is a body fluid. Agree? It is a body fluid. So, apart from genital fluid, I can make it a column, right? That will be much more better. So, I'll write it over here for you guys. We have body fluids and not all body fluids. Okay, our genital fluids. And then comes blood. So, it's saying either transfusion of blood or basically blood products, whatever it is, okay. Next thing, by sharing infected needles, as in the case of intravenous drug abusers. So, people who take drug with the help of intravenous methods, okay, they are injecting things. So, at this point of time, and people are sitting together and they're sharing the needles, and if one among them have HIV, okay, obviously blood is there on the needles and you're injecting it in the other person also. So, again things would be spread. Okay, so that is the third thing. Now, from infected mother to her child through placenta. So, when baby is born, you know, when baby is born, inside the mother's womb, there is something that is known as placenta, which provides nutrition and helps in exchange of gases to the baby from the mother. Now, how it happens, it is a big question. It's a big question mark. But the thing is, there might be some, usually the blood of the mother and the baby does not mix. Okay, usually our blood and our mother's blood do not mix when we are in her womb. But there are rare cases in which the placenta could be having certain tears or inflammation such that the blood transfusion could take place. In such cases, there are high chances that basically you could get the disease. Okay, so it can be from from the mother to child via placenta. Well, apart from that, it also happens, it is not there in your NCERT textbook, but let me tell you, it also happens while breast feeding if the mother has HIV virus. Now, this last two portion, that is mother to child as well as breastfeeding, basically I have read this, that is when antiviral therapy is happening, and if mother and the child both are given equal treatment for that, then there are lesser chances for the spread of the disease while these two methods. But yes, if not taken care of, obviously this is a big red flag. Okay. So, we have understood from the whole paragraph what do you mean by AIDS is acquired immunodeficiency syndrome. That is something that you are acquiring through your lifetime, and in this particular disease your immune system becomes very deficient, very weak. There are so many symptoms that it starts to show. Now, it is caused by a virus which is known as human immunodeficiency virus, which comes in the group of viruses which are known as retroviruses or old viruses. You can think like that. And these viruses, no children, they have RNA as genetic material. They do not have DNA. They have RNA as genetic material, and also they have envelope. So, we say that these viruses are enveloped in nature, they are covered in nature. Okay. Going ahead, how can anyone get this particular disease? First thing, sexual contact with infected person. Second, transfusion of contaminated blood. Okay. Third thing, from infected by using or by sharing infected needles while having intravenous drugs. Okay, that is one thing. And the last thing is basically from mother to child through the placenta, and also through breastfeeding, which I explained. But again, as I said, when antiviral therapy is done, okay, it is made sure that the child is not affected much, because you know, breastfeeding and all, everything, when the milk goes on, it does have HIV viral proteins. But the thing is that when antiviral therapy is given, the chances for it becomes very less. Okay, and you know, if it's a baby, there are high chances for it to have developed the disease, because it does not have a strong immune system. So if taken proper care, probably the chances could be made less. So can we write this now? Okay, acquired immunodeficiency. I don't want you to mug this up. Understand this thing, okay? Syndrome. Is it congenital, children? Is it congenital? Is it genetic? Does it go and mess up your genes and then you will inherit it and things like that? No, it is not like that. It spreads through the body fluids, especially blood, and basically the genital fluids. Okay. How is it spread? As I said, blood transfusion, sexual contact, sharing of needles, mother to child via placenta. Okay, and usually, basically, this is from genital fluids. And tell me, genital fluid and what? Blood. Okay. Who are at risk? Tell me, who are at risk? Basically, who have multiple sexual partners, they are at risk. Drug addicts, they are at risk because they are taking intravenous drugs, okay, if they are taking intravenous drugs. Second thing, who require repeated blood transfusions, and children born to an HIV infected mother. So basically, over here, blood transfusion, basically diseases which require blood transfusions. Okay, so people who have diseases that requires blood transfusion, what are those diseases? We'll come to that, but I'm just writing. Okay, sexual contact, when it becomes a problem, obviously when it's a HIV infected person, and also when you have multiple sexual partners, is increasing the risk. Who are at risk, they are asking. Okay, multiple sexual partners. Okay, and then drug abusers, they are at risk, and obviously mother to child, which is just there. Okay, so that is there. Now this and this would not be a fault of a person if they do not know it priorly. Okay, but This and this, obviously you have to be much more careful for that. Okay, now that can also happen accidentally, but still that is what is written in your NCERT, and I'm just teaching you that. Now going ahead, okay, one more thing. How is it not spread? How is it not spread? It does not spread through saliva, children. It does not. Okay, does not spread through saliva. So while I was teaching in the class, one child asked me, "Ma'am, is it spread through kissing?" Well, the problem is our mouth can have bleeding. It can be gum bleeding, or we can have mouth ulcers. Now at this point of time, what happens, children? The blood from our mouth mixes with the saliva. Agree? At this point of time, again you are risking the chances. So naturally it is not spread, but obviously if the person have bleeding gums or something like that, it could be spread. So have this consciousness as well. Does not spread through saliva when it is not mixing with blood by any chance. Okay, it does not spread with tears. It does not, okay, that is also important. Does not, how doesn't it spread? It doesn't spread when you are having a handshake. Tears, saliva, fecus, no, nothing like that. But again I am telling you, while handshake, okay, that person is having a bruise, okay, some very open bruise, and you also have it, and then there's a contact, that's when things can happen. Okay, that is one thing. So again, as I said, it's not like you have to avoid people who are having HIV. There are so many people who are having HIV and they are living their life very decently. So basically, and it's not like everyone does something very wrong that they get a disease. It's not like that. There are so many things that could happen. So I had a neighbor, okay, in my town who got this disease because they went to a parlor where the needles that they used, not the needles, basically the, what do you call it, blade that they used was reused. Now they got it something like that. So basically, can you blame them for that? Obviously you should be going to a proper place, that is something different, but can we blame a person for that? No. Okay, so that is one awareness that we should be having, that it's not their very big fault or they did something really bad that they got it, and we should not be avoiding them as such. We should also be greeting them just as normal human beings do. Just be aware of what you should not be doing and be aware of who are at risk. Okay, these things probably, if you are aware of it, then you will be more conscious while doing things. That's all. Okay, going ahead. Do you know when do people need repeated blood transfusion? I was talking about, right? Find out and make a list of such condition. So diseases that needs blood transfusion, children. People who have sickle cell anemia. People who have sickle cell anemia. People who have hemophilia, where the blood does not clot and it keeps going on. People who are having thalassemia. Okay. So all of them, they need blood transfusion and they have to be really careful while doing this. So now usually blood banks have a very safe mechanism of keeping the blood. They check whether the person is infected with some other disease and then only they keep it, but still. Okay. Now it is important to note that HIV/AIDS is not spread by mere touch or physical contact. It spreads only through body fluids. It is hence imperative for the physical and psychological well-being that HIV/AIDS infected persons are not isolated from family and society. I know people who stay very peacefully because people around them are really nice. So let's also be an example of nice people, and let's also greet them as you greet any of your friends. Now there is always, infected person, there is always a time lag between the infection and the appearance of AIDS symptoms. Okay, there is a time lag, and the time lag could be from a few months to many years, usually 5 to 10 years as well. Now why is that so? Because HIV virus basically infects our cells, gets inside our body and is going to affect some cells of our body which are known as T lymphocytes majorly. Majorly they are going to affect T lymphocytes. Now these T lymphocytes, when they start affecting, it's not like we have just one T lymphocyte, no. So the time comes when they are majorly affecting a huge lot of them. that is T of lymphocytes, and it takes a huge lot of years as well. That's when we start to show the symptoms as well. So it's not a one-day process. The HIV has to get inside our body, inside our cells, replicate itself, kill all the T lymphocytes which are being present over there. Not all, at least majority of them, and it will definitely take time. Some people might get it earlier. The symptoms could show up earlier. Some people could show it up very late as well. Okay. Okay. Now. Pathway of the infection. Now this is the most important thing. I think that we should be making the story of HIV, and then we shall talk about the pathway. So children, as you can see here, as I promised, this is HIV. Hi, I am HIV. Okay, so over here you have to understand the structure of HIV. So basically, this particular envelope that you see in blue, light blue in color, okay, I can mark it up and then I'll remove it because it won't be seeming really nice. So this particular green thing that I am making over here, okay, what is it, children? It is the envelope, okay, and it is made up of lipid. It's lipid. Okay, just a second. I'll make a bit down because we have to make some things there. So it is lipid envelope, and it is made with the help of our cell membrane itself, which is why people do not note it really well. It is made up of our cell itself, cell membrane itself. I am going to tell about that as well. Now over this you see these proteins which are attached, which looks like lollipop, right? What are those things? They are known as GP120. Okay, what do you mean by GP120? GP120 means glycoprotein 120. Now these glycoproteins are very much important because these are the ones that are going to attach to the receptors on the cell. What cell? We'll come to that. Now, so envelope is done, lipid envelope is done, then glycoproteins are also there, 120. GP120 is also there. Inside this, if you see, there is a structure like this which is known as, okay, something I should not be drawing here again. Yeah, this structure that you see, it is known as capsid. Okay, what do you call it? Capsid, that's going to protect what? The RNA which is over here. So over here that you see something, it is RNA, ribonucleic acid. Now this RNA, children, you have two of them. Even though in this picture there is only one, I'll make two. Okay, you have two single stranded RNA. Okay, there are two single stranded RNA, not double stranded, they are single, separate strands. Okay, and apart from that, you might be seeing that there are some green, red and things, some structures like that. I could zoom this in and show it to you. Just a second. So over here you could see some structures like this. What are these structures, children? These are enzymes. These things that you see over here, these are enzymes, and where should I write it? I'll write it here. Okay, these are enzymes, and the enzymes, the short form of it is RIP. What do you mean by RIP? Not rest in peace, it's something else. Okay, so basically we got to know few very important things, and this is very important for your NEET exam as well. They ask so many questions regarding the structure. You have an envelope. On the envelope you have GP120, that is the glycoprotein. The envelope is lipid envelope. It is made from our cell membrane itself. And now inside, if you see, they have a capsid which protects their RNA. How many RNA they have? They have two single stranded RNA. They have two single stranded. They are not double stranded. They are not together. Okay, they are two single stranded RNA, and then basically they also have enzymes within them, which is RIP. What does RIP stand for? I'll tell you. Before that, let me just turn on the AC because it's too cold. Che, too hot. Yes. Now, this HIV virus gets inside our body with the help of blood or whatever, our sexual fluids, the genital fluids, and now it is going to affect. Okay, I think this could be enough. So this is a cell. Let me name this cell as T lymphocyte. Okay, What T lymphocyte? TH. It is TH cell, okay, T helper cell. Basically, this virus comes over here slowly, and on the membrane, no, children, okay, on the membrane we have a protein which is known as CD4, okay. TH cells, basically they have a protein, it's there in other cells also, that is what? CD4. Now GP120 comes and sees this, oh, it's fine, this is the same thing that I wanted to bind on this. So basically this whole virus turns around and comes over here, and the GP120 binds over it, and we have the whole structure as well with the capsid as well as the viral RNA and the enzymes as well. Now when they bind, they slowly fuse with it. When they bind, they slowly fuse with it such that they just have the outermost portion with the GP120 over here, but they have their RNA inside and also their enzymes, okay. Now what will they do with the RNA? So basically inside our body, when we say that how is protein made in our body, it's something like this, where basically the DNA which is there, it gives information to make mRNA, that is messenger RNA. Now this messenger RNA goes outside our nucleus and it makes proteins with the help of ribosomes. But here something reverse is happening. Here RNA is going to make the DNA, and that is where the enzymes role come. And what is the first enzyme that we are having? The first enzyme that we are having is reverse transcriptase. Reverse transcriptase. So this process of DNA to mRNA formation, what do you call this usually? You call this process as transcription. You call this process as transcription. Okay, but now here ulta is happening, exactly opposite happening, where an RNA is going to make a DNA, which is why we say that transcription process is reversed, and the enzyme which is used is known as what? Reverse transcriptase. That's not all. That's not all. It says, since I am inside your body, I want to be just like how your genetic makeup is there. And how is our genetic makeup, children? If you see our genetic makeup, we have double-stranded DNA. What do we have? We have double-stranded DNA. So we have two strands of DNA which are complementary to each other and they coil around. So this particular guy feels alone, like I just have a single DNA, I don't want that, I want one more DNA. Basically there will be one more DNA that is formed with the help of same enzyme, and now they will form double strand. Now this one single DNA has now become double-stranded DNA. Double-stranded DNA with the help of same enzyme, that is reverse transcriptase, and this double-stranded DNA is now taken inside the nucleus of our cells, and basically there will be another enzyme which will be active over here, and that enzyme's name is integrase. Now as the name suggests, it is going to integrate this particular DNA into our DNA. And how can you do that? Basically integrase cleaves our DNA, okay, integrase cleaves our DNA, and basically what will happen? We will have some structure like this, okay, and in between that what would we have? We will put the DNA which is made by the virus. right? And this DNA obviously will be having all the information about what does the virus need, about what proteins the virus has to make. Probably this GP120 protein, or probably whatever enzymes it has to make, everything would be now made with the help of this particular DNA. Because in our body, as I said, DNA usually gets transcribed to mRNA, and then mRNA gets translated to proteins. So same thing happens over here. This particular DNA, along with the whole DNA that we have, basically it gets transcribed, and what do we have? We have mRNA being formed over there. This mRNA with the help of endoplasmic reticulum, okay, I'll make it in different color. Okay, this mRNA usually goes to endoplasmic reticulum, and with the help of ribosomes, which are there, it forms what, children? Viral protein. Okay, now obviously they have to make a lot of proteins, and for that they have to cleave themselves so that different parts could be utilized. So for that, the virus has the third set of enzyme, which is known as protease. Third set of enzyme, which is known as protease, which is going to act on this viral protein and make it smaller chunks, make it peptides. So first this one, second this one, and third one is this, where the whole big viral protein is broken down to smaller chunks, which is known as peptides. So these proteins are broken down to peptides, and these peptides are going to make each of the parts. So for example, the GP120 is made. So this GP120, after being made over here, they will slowly travel down and come over here and be formed like this. They will come and attach like this, and then obviously what will happen? Basically, again, where was I? Apart from this, there will be enzymes that will be made. Agree? Do you agree to me, children? Yeah. And obviously RNA also you can make from DNA. So your RNA, which was there initially, that would also be made over here. So this particular DNA can make RNA out of it, and this particular DNA can make mRNA, which is going to form what? Viral proteins as well, everything. So your whatever is needed by the RNA is all set. It has its own RNA back again, and then it has its enzyme. It also has the the glycoprotein 120, and it also makes the proteins needed for capsid formation. Okay, so it has the capsid also now. Everything is there. So what happens? Slowly this area will pinch off, such that you will get something like this, where you have capsid, where you have RNA, where you have your enzymes as well. And this particular virus is going to affect the other cells as well in the same manner. Okay, I'll just repeat it once, then I'll come to your doubt, which I know probably what all you would be having. Initially, we have the structure of our virus. This virus says I can only attach to a receptor, that is CD4 receptor, and it attaches with a protein, that is glycoprotein 120, which it has it by itself. Goes and attaches there, gets inside. Okay, the membrane fuses and lets everything inside, that is the capsid, the basically your viral RNA, your enzymes, everything. Okay, now the viral RNA is inside along with the enzyme, that is reverse transcriptase. So usually transcription means making mRNA out of DNA, but here the opposite is happening. RNA is going to make DNA. So RNA is going to make DNA, but then DNA is like, I cannot be alone, I want a partner. So basically one more strand, complementary strand is made, which is known as DNA double strand, okay, by the same enzyme itself. Now this double stranded DNA is now going to get integrated into our DNA. Because it wants to use our machinery. Okay, so you know our DNA is going to make mRNA and then proteins. So same machinery can be utilized by the virus as well. So they use our machinery to form mRNA. This mRNA, when read by the ribosomes, gives out viral protein, and the viral protein later, it's a big molecule, so you have to break them down into peptides. That is done by protease, which is the third enzyme. So which is why RIP is there. Now slowly each protein, everything is made. You get glycoprotein, you got the enzymes, which are also proteins, you got the viral RNA because it is made by the DNA itself. The DNA can convert into RNA. So your RNA is made, your capsid is made, everything is made. Slowly they chip off from the whole cell and they move around to different cells. So the question that you might be having is that, ma'am, what is our body doing at this point of time? Our body obviously knows that there is a problem. Okay, it can sense the problem, right? It can sense the problem, it can find the problem, then why is it not doing that? Basically because, usually when there is some problem, our cells basically cleaves whatever proteins comes in and present it with the help of some other proteins which are known as MHC, major histocompatibility complex. But at this point of time, they are also cleaved, they are also managed in a way that they are said that come inside. So no signs of attack is shown by these cells that are affected by the virus. In this way, they go on repeating the same thing. Now, I want to tell you one thing, that is usually the cells which are affected are macrophages, the phagocytotic cells, okay, the macrophages. Slowly, they start affecting the T helper cells. Now, T helper cells are the one that helps the whole humoral system as well, humoral immune response that you have studied. They call the B cells to produce antibodies and everything. TH cells are involved in killing the other cells as well, indirectly. So basically, this particular virus is going to affect a major part of TH cells, which would obviously lower down our immune response. So initially macrophage, and then it goes on affecting TH cells, and they have different things. Basically, when the virus affects the macrophage, okay, it stays there and the virus replicates itself, okay, which is why macrophage is known as HIV factory. It is not very easy to kill the macrophage. It takes time. So it becomes the HIV factory for production of more and more HIV viruses. Whereas when it goes and affects the TH cells, it gets killed. It gets what? Killed itself. So this is not known as the HIV factory. Macrophage is known as HIV factory. This is where the HIV viruses replicates in a more amount. Okay, it sustains for some time. But when it goes and affects the TH cell, it is going to kill them. And one more thing, it is actually searching for TH cell only. This is something that comes in the midway, that's all. So it is actually wanting to affect the TH cell, because if TH cell are affected, that's when the immune response will be really less. Okay, so that is one very important thing that I wanted to tell you, and this is the story of HIV. I hope you got it. Now, yes, let's read it. After getting into the body of the person, the virus enters into macrophages. As I said, the first thing that they do is enter into macrophages, where RNA genome of the virus replicates to form viral DNA with the help of the enzyme reverse transcriptase. This viral DNA gets incorporated into the host DNA with the help of integrase. Okay? And directs the infected cells to produce virus particles. The macrophages continues to produce virus, and in this way acts like a HIV factory. Simultaneously, HIV enters into helper T lymphocytes, which are very much important, replicates and produce progeny viruses. The progeny viruses released in the blood attack other helper T cells. I told you that this is the primary motive. They want to kill the helper T cell, because this is the one that is very much strong, and they are the one that is responsible for a whole immune response. So if you killing them, your immune response goes down, and this particular cell viruses can be really safe in there. This is repeated, leading to progressive decrease in the number of helper T lymphocytes in the body of the infected person. During this period, the person suffers from bouts of fever, diarrhea, and what? Weight loss. So you have to understand this. Fever is there, diarrhea is there, weight loss will also be there. Fever, diarrhea, and weight loss. Okay. Due to decrease in the number of helper T lymphocytes, the person starts suffering from infections that could have been otherwise overcome, such as those due to bacteria, especially Mycobacterium. That is, one example of this is what? Tuberculosis. Okay. So basically, your immune system is weak. At this point of time, you can get several diseases, whatever is it. So it can be mycobacterium based disease, that is tuberculosis, lepre, whatever. Then you can have viruses being attacking on you, fungi attacking on you, and parasites like toxoplasma. Okay, parasites like toxoplasma, which affects your brain. So basically, these parasites could also affect you, but usually it is overcome. Usually when we are healthy, it's overcome. But people who are having HIV/AIDS, they are most likely to get everything, because their immune system is, children, shut down. Okay, the patient becomes so immune deficient that he or she is unable to protect himself or herself against these infection. A widely used diagnostic test for AIDS is enzyme-linked immunosorbent assay. What is it? Enzyme-linked immunosorbent assay. So I'll write it over here or here. Enzyme-linked immunosorbent assay, where what do they do is they have a tray where with the antibodies being present, and then they put the antigen of a person who is probably infected with the virus. Now the antigen can come, if it is matching this particular antibody site, it will come and attach, and then they are again putting an antibody. They are again putting an antibody which will go and bind to this particular antigen itself, if the antigen is in the bonded state. If it is in the free state, obviously the antibodies won't be coming and attaching, but if it's in the bound state, the antibodies will be coming and attaching to it. Okay, now this person has what? HIV/AIDS, which is why antigen came and attached, and then later on the antibody also came and attached. Now this antibody is linked to an enzyme. Okay, which basically converts a substrate. Okay, for example, after this is done, a substrate is being put, and now with the help of this enzyme, if the substrate color changes to something else, which means that this particular person is having HIV. This is what you mean by enzyme linked immunosorbent assay, because here you are using an enzyme to detect the presence of the particular virus. I'll tell you again, there is antibody present over here, onto which you are putting the suspected person's antigens. Okay, if it comes and binds, it will bind there, and now once it binds over there, okay, there will be another set of antibodies which are linked with enzyme that are given. Now when it also goes and binds there, and when we put the substrate and the substrate color changes, that means probably this binding has taken place, that means the person will be having HIV. We will be studying about this in another chapter. Okay, as of now, just a small gist of it that I told you. Okay, if you want to know more about it, you can tell me in the comment section that ma'am, we want a 10 minutes or 5 minutes video. I can do it also. Okay, it's not a problem. Now going ahead, treatment of AIDS. So please remember the name, it is ELISA. It is what? ELISA, enzyme linked immunosorbent assay. Going ahead, treatment of AIDS with antiretroviral drugs is only partially effective. So obviously treatment can be done, but is it curable, children? It is not curable. Okay, it will be partially effective, but it is not curable. So antiretroviral drugs are being given, antiretroviral therapies are being done so that the person could survive. But obviously, as I said, it won't be curing the disease. It can increase the longevity of the person's lifetime. That's all. Okay, so as they are saying, they can only prolong the life of the patient but not prevent the death, which is inevitable. So with this particular information, I have few things to say. Okay, so the HIV factory is macrophage. Okay, the HIV virus are going to attack macrophage first, and then they are going to attack the T cell, that is T helper cell. There are different variety of T lymphocyte, children. T cytotoxic cells are there, T memory cells are there. As of now, we are talking about T8 cells, that is T helper cells. Okay, please remember that. Now, if a person has that, okay, then the initial symptoms will be fever, diarrhea and weight loss. Then later on, they will be contracting lots and lots of diseases which they probably could avoid or overcome if they were normally healthy. Okay, if they were normally healthy. So over here, it could be toxoplasma, which is a parasite which could be affecting the person. Okay, it could be Mycobacterium tuberculosis, anything, or any other virus or any other fungus which could affect them very easily. So for now treating them, we have antiretroviral. Retrovirus is the name of the virus, right? Or the family of the virus. Basically, anti as in against it. So antiretroviral therapy is there which could prolong their lifetime but would not cure the disease. Please understand that HIV is not curable. Now next thing, how do we diagnose it that the person has HIV? By doing ELISA, ELISA, that is enzyme linked immunosorbent assay. Okay, now going ahead, what do we have? Okay, let's read. Human immunodeficiency virus has a protein coat and a genetic material which is, tell me, what is the genetic material, children? I taught you. Answer down in the comment section. Tell me what is it. Okay, you put your answers, I'll be explaining it to you at the end. Okay, so usually I give it to you but this time I want you to do it. It is double-stranded RNA, double-stranded DNA, single-stranded DNA, single-stranded RNA. You tell me in the comment section. You can pause and find the answer and tell me. Now going ahead. Prevention of AIDS, as AIDS has no cure. Okay, so prevention of AIDS is the topic. AIDS has no cure. Prevention is the best option. Moreover, HIV infection more often spreads due to conscious behavior patterns, and it's not something that happens inadvertently. That means people, it happens due to conscious things. We are aware that we might get these diseases, but still we do not take a possible effort to avoid this. Okay, that is what they are saying. Like pneumonia or typhoid. Okay, so it does not happen unknowingly like pneumonia or typhoid. That's what they are trying to say. Of course, infection in blood transfusion patients, newborns, mother may take place due to poor monitoring. So that is not a person's fault. It can happen due to poor monitoring when it is spread through blood transfusion as well as from a newborn from the mother. So it takes place through poor monitoring. The only excuse may be ignorance, and it has rightly said, do not die of ignorance. In our country, the National AIDS Control Organization, NACO, you have to remember the name, okay? NACO, National AIDS Control Organization, and other non-governmental organizations, that is NGOs, are doing a lot to educate the people about AIDS. WHO, that is World Health Organization, has started a number of programs to prevent the spreading of HIV infections. So that I have written over here, blood from blood banks safe from HIV. So this is one initiative that they took, basically WHO. Okay, so blood that is from blood banks would be safe from HIV. They have to make sure of that. Okay, second thing, ensuring the use of only disposable needles and syringes in public and private hospitals and clinics. So no one is allowed to use one particular needle repeatedly. You have to dispose that and use a new one. Okay, that is one, so that the blood transfusion, the method through which it could be transferred with the help of needles and things like that, it could be avoided, and by blood transfusion also it could be avoided. Next thing, free distribution of condoms, because if condoms are used, it acts as a physical barrier against all the STIs. Okay, so basically free distribution of STIs are also, free distribution not of STIs, children, condoms are also done to prevent spreading of these diseases. Now, what do you mean by STIs? Sexually transmitted diseases. Okay. Now, controlling drug abuse, that is one big thing, because people still use syringes for intravenous drug injection. You should not be doing that. If you are controlling it, probably everything would be sorted, and they are also trying to control it. Okay. Next thing, advocating safe sex and promoting regular checkups for HIV. So they are also asking, you might have seen ads for it. HIV is not a taboo anymore. So they are promoting that you should be having a regular checkup for all those things. So this is how basically we are trying to combat the disease, to combat the spread of the disease, and to have a very safe life. Okay, so let's read it again. So as I said, ensuring the use of only disposable needles and condoms, and controlling drug abuse, advocating safe sex are some checkups for HIV in susceptible population are some such steps taken up. Infection with HIV or having AIDS is something that should not be hidden. Okay, if it is hidden, then probably you are consciously trying to spread it to other people as well. That is also wrong. Okay, since then the infection may spread to many more people. HIV AIDS infected people need help and sympathy instead of being shunned by the society. That is for us. Okay, we should not be making them ashamed in our society. We should be providing them support shoulder to shoulder, right? They are just like us. Unless society recognizes it as a problem to be dealt in a collective manner, the chances of wider spread of disease increase manifold. It is a malady that can only be tackled by the society and medical fraternity acting together to prevent the spread of the disease. So if we act together, then only this disease can be eradicated from our world. Okay, so basically that is there. Please remember NACO, National AIDS Control Organization. National AIDS Control Organization, how can you remember it? Nationally, you are trying to control AIDS. National AIDS Control Organization, NACO. Okay? Okay. HIV that causes AIDS. Now we can solve questions, right? So over here, I am not solving it over here. We will solve it together over here. Chalo. It's visible? Yes. HIV that causes AIDS first starts destroying what, children? Among them, what is the first thing that they start destroying? Usually it is what? Macrophages, but macrophages is not there. Macrophages is not there, which is why it is a cancelled question, I hope. But among them, helper T lymphocyte, thrombocyte, B lymphocyte, leukocyte, it is what? Helper T lymphocytes. It has to be macrophages because that is the first thing that is getting affected. But since it is not there, helper T lymphocytes. Human immunodeficiency virus has a protein coat and a genetic material which is, you told me in the answer section or what? Okay, what is that particular genetic material that they have? They have double-stranded RNA. Is it that? Is it that? Since I said there are two RNA, that does not mean that is double-stranded. I said that they are single, separate strands. So it's single-stranded RNA, children. It is what? Single-stranded RNA, but two of them are there. That's the only difference. Please understand the difference. So it is single-stranded RNA, but they are in two in number. Which of the following is correct regarding AIDS causative agent HIV? HIV is enveloped virus containing one molecule of single-stranded RNA and one molecule of reverse transcriptase. Is it like that? Second thing, HIV is enveloped virus that contains two identical molecules of single-stranded RNA and two molecules of reverse transcriptase. HIV is unenveloped retrovirus. That is completely wrong. HIV does not escape but attacks the acquired immune response. Does it escape? It is said it does not escape. HIV does not escape but attacks the acquired immune response. What is the acquired immune response? Basically T lymphocytes and production of antibodies, all this thing. So they are not escaping and they are attacking the acquired immune response. This is entirely correct. What about this? HIV is enveloped virus that contains two identical molecules of single-stranded RNA. This is also correct. And two molecules of reverse transcriptase. That is a debatable thing. But this is correct. Okay, this is comparatively more correct. In that year, both these answers were given right option. But preferably going with the option because it cannot be wrong. Okay. Now, at which stage of HIV infection does one usually show symptoms of AIDS? Within 15 days of sexual contact with an infected person? No, it can take up to lots of years as well, right? Okay. When the infected retrovirus enters host cells? When the infected retrovirus enters host cell, when HIV damages large number of helper T lymphocyte, when the viral DNA is produced by reverse transcriptase. Tell me, when do we start showing symptoms? When it enters our body, is it? Is it like that? Or when it starts to produce DNA, is it like that? No. When it starts to damage the large number of helper T cells, that's when it starts to show the symptoms. I told you that as well. Okay. Which one of the following statements is correct with respect to AIDS? HIV can be transmitted through eating food together with an infected person? No. Drug addicts are least susceptible to HIV infection. They are more susceptible. AIDS patients are fully cured 100% with the proper care and nutrition. Wrong. The causative HIV retrovirus enters helper T lymphocytes, thus reducing their numbers. That is correct. They enter, they kill, they reduce the number. Okay, it is not spread through eating food together. Lots of people who eat food together do not get the disease. As I said, you should not be doing any such activities where blood transfer is there. Apart from that, everything is all good. So you don't need to worry about it. So these were the 5 questions. I hope you were able to do that as well. Now, one or not just one question. There are 2 questions that I have for you today. They are easy, which is why I kept 2 in number. HIV enters T cells. Which one? Death in AIDS is due to what? So you can take a screenshot of this, or you can find the PDF in the description box as well. So that's all, children. I hope that you were able to understand whatever I taught today, and I just wish you all the very best for your exam and your preparation. Just be happy, do not worry, and let me know if I am helping you really well. And please do not forget to subscribe to the channel because we are working a lot to make things very easy for you. And the next session will be on cancer. Do not miss it. Bye-bye. Take care.